Good day to doctors. I'm Ernie with Matrix ID BSB3113. I'd like to explain why is it important to use the genome editing system to in the human malaria parasite, which is the Plasmodium falciparum that had in the female mosquito Anopheles. Based on the previous research, it is to discover that over 600,000 of the people die annually is just because of getting infection of the malaria. The enormous amount of people dying due to the malaria infection is an issue that we really need to concern about it, and that's why a genome editing system such as CRISPR-Cas9 is important to be introduced to this topic so that to tackle and combat these malaria infection issues. Through this genome editing system, such as CRISPR-Cas9 system, it helps us to develop better understanding toward the parasite biology and develop a novel strategy to combat with the malaria disease. This technology is important as it can accurately and precisely genetically modify the strain that enable the mosquito to not release the causative agent, which is the Plasmodium falciparum, that will harm and induce the malaria in the human body system. Some more, this technology is also good because it is less virulent to both parasite and human immune system and contribute much to control and eliminate the malaria. Now I'm going to explain in details on how the principle of the CRISPR-Cas9 system work on the human parasite malaria. So the first thing in what you need to do by using the CRISPR-Cas9 editing system is you need to firstly identify a targeted gene that we want to modify. And in this case, the targeted gene is a chromosomal EGFP locus in the Plasmodium falciparum. After that, we need to design a sgRNA, which is known as a single-stranded guide RNA, specifically for the EGFP gene. After that, we need to construct the plasmid that is specifically for the targeted gene, where the plasmid must contain the guide RNA as well as the Cas9 enzyme. Once the plasmid is done for construction, the plasmid will be introduced into the parasite system, and the CRISPR-Cas9 protein will subsequently interact with the sgRNA to activate the CRISPR-Cas9 system. After the activation of the CRISPR-Cas9 system, the guide RNA will guide the CRISPR-Cas9 protein to reach the specific location and bind to it. When the CRISPR-Cas9 protein binds to the specific location with the targeted gene, it will create a double-stranded break, which is known as BSB, by cutting the two strands of the DNA at the specific location of the genomic. So this is the purpose to create the BSB is to disrupt the origin sequence of the targeted gene so that to eliminate the causative agent that will cause to the malaria disease. Once the BSB is formed, we need to repair the BSB damaged region through a homology direct repair mechanism, which is known as also HDR mechanism. While in this mechanism, it will utilize the DNA sequence homology to accurately repair the BSB damage at the correct genomic location. The HDR pathway protein will recognize the homologous sequence of DNA near the region of the BSB. Once the homologous repair is done, a PCR analysis is conducted to ensure that the causative agent inside the gene is fully been disrupted and eliminated, so that when people get bite by the genetic modified mosquito, they won't get infection to the malaria disease.
First, I will going to present about the advantage of the technology. The first advantage is precise and targeting. CRISPR-Cas9 is highly precise targeting of the specific DNA sequence within a genome. Two single-guided RNA strands are designed to guide the Cas9 polypeptide to cut the two areas, and the genes of interest was removed. The second advantage is high efficiency. CRISPR-Cas9 is highly efficient in inducing targeted genetic modifications. The Cas9 enzyme efficiently introduce double-strand breaks in the DNA. Genome editing with CRISPR-Cas9 harness cellular double-strand break repair pathways such as non-homologous end joining. Thirdly, permanent gene knockout. Gene is modified permanently. The CRISPR-Cas9 gene editing technology is the ideal tool for the future for the trending disease by permanently correcting deleterious-based mutations or derusting disease-causing genes with great precision and efficiency. Lastly, reduce the off-target effect with optimizations. By using a special algorithm, CRISPR-Cas9 can achieve reduced off-target effect. Off-target effect can be minimized, and the result of the experiment will be good. Next, the advantage of the technology. Off-target mutations is a key limiting factor and has become a mainstay in effort to improve the CRISPR system, particularly in the production of transgene-free crop. Off-target genome editing refers to the DNA modifications at unintended and not specific site and can occur by guided RNA or in a guided RNA independent manner. Second, efficiency and specificity variability. The efficiency and specificity can be varied among different target site and cell type. Thirdly, is the delivery limitations. In order to produce the required modifications, CRISPR-Cas9 must be successfully inserted into the cells. Delivery of the Cas9 guided RNA compressed into a sufficient number of the cells can be difficult, like the many other gene editing applications, because each component needs to be supplied into the cells at the correct concentrations and at the correct stage of the cell cycle. The delivery of the CRISPR's reagents can be achieved using lipofection, erythroporations, microinjections, nanoparticle, and viral plasmid. Lastly, the safety and commercialization issue. The adoption of the genome-edited crop is a dilemma. Some countries label the products of this technology as transgenic-free, and some countries still debating on this issue.
Hi, my name is Tan Chun Chun. Now I'm going to talk about the ethical issue of genome editing in the human malaria parasite Plasmodium falciparum by using the CRISPR-Cas9 system. The ethical issue of this genome-edited parasite to our environment is if it is introduced into our environment, there might be concern about the unintended consequences and ecological impact. For example, the risk of transferring gene to other species, such as passing the negative traits to related organisms through mating. Next is the ethical issue in human. When the human cells or tissue are involved in the research, such as host-parasite interaction, without obtaining any informed consent, it will be illegal to proceed with the research. This is the ethical issue in economy, like commercialization and intellectual properties. The problem is concern about accessibility and affordability arise. For example, if essential technology or treatments are patented, there is limited access for the lower income population.